Kita discuss sampai mana guys?
Transcription.
Transcription lah. Kiranya saya dah discuss proses initiation, elongation semua tu
Dah.
Okay. Alright. Genetic code dah ke? Genetic code dah ke?
Belum.
Alright. Untuk genetic code belum. Okay. Okay, so tapi kita dah discuss transcription. Okay, kita dah discuss transcription. So just to recap on previous lesson, for transcription you already learned the process. Okay, what happened during transcription. So before we recap on previous lesson, I just want to ask you, okay, where does transcription occur? Where does transcription occur?
In the nucleus.
In the nucleus. Thank you, Adele. All right. Then we have transcription. We have 3 stages. What is the first stage? Farah, what is the first stage, Farah?
Initiation.
Initiation. Very good. Shasha, second stage. Shasha, what is the second stage, Shasha? Oh, Shasha, internet problem. Okay, it's okay. Falih, can you tell me what is the second stage?
Elongation.
Elongation. And last one, we have stage called Hisham, apa dia? Hisham ada tak? Hisham tak masuk lagi. Ife. Apa last stage Ife?
Termination.
Termination, very good. So for transcription, we need promoter, RNA polymerase, and we need terminator. And the product of transcription is mRNA. What happened to this mRNA? Dia akan pergi mana mRNA ni? What happened after mRNA is produced? Sorry?
Dia akan pergi cytoplasm.
Okay, it will go to cytoplasm. Untuk buat proses apa? Imani. Imani, ada tak? Imani?
Ada madam.
Proses apa? Proses apa? Saya tanya apa tadi? Yes, proses apa? After mRNA go to cytoplasm, dia buat apa?
Translation.
Translation, very good, pandai. Apa dia? Apa Imani? Kau nak cakap apa? Tak ada apa. Okay. Alright, before we proceed on translation, I'm going to explain about genetic code. Okay, genetic code and its relationship with the sequence of amino acids. So nanti saya akan bagi quizzes. Okay, saya akan bagi quizzes on transcription and translation 20 soalan. Saya akan tanya pattern soalan yang macam ni. Okay, nanti awak punya UPS pun ini adalah pattern soalan yang selalu ditanya. Okay, so I'm going to explain genetic code first. Okay, so this is the diagram. You can see that the DNA is double stranded. Okay, it runs from 5' to 3', and then from 3' to 5'. This characteristic of DNA we call that as apa? Siapa nak Isyam nak join? Okay, kita panggil DNA 5' to 3', 3' to 5'. Apa karakteristik DNA yang kita panggil macam ni?
Madam.
Ya.
Madam share screen ke?
Ya.
Memang tak nampak.
Yang lain nampak ke?
Tak nampak, madam.
Serius lah? Ya ke? Thank you bagi tahu. Ke saya tak share pun? Ya Allah. Ya Allah, saya tak share pun. Kejap. Yelah 8 pagi, maafkan saya. Sekejap. Ini dia. Barulah ada. Okay. Patutlah tak keluar dekat handphone saya. Okay. Sekejap Okay, nampak kan? Nampak tak?
Nampak madam.
Alright, so this is genetic code. DNA is anti-parallel. Okay, saya cakaplah terus DNA is anti-parallel. Maksud anti-parallel, dia run from 5 prime to 3 prime. 3 prime to 5 prime. And DNA is anti-parallel is important for the process of DNA replication. Okay, for protein synthesis, we're going to transcribe DNA from DNA to mRNA. Maksudnya kita akan copy, kita akan transcribe, okay, kita akan transcribe DNA from DNA to mRNA. So we have double stranded, okay, we have non-template strand and DNA template strand. Okay, this one we have two strands, DNA template strand and non-template strand. Okay, so my question is, which strand that we going to copy? Strand yang atas ataupun strand yang bawah? Which strand that we going to copy?
Yang bawah, madam.
Why yang bawah?
Sebab nama dia template.
Alright, very good. Katakanlah saya tak tulis DNA template strand ni, tak ada DNA template strand. Okay, for example, I remove this one. Okay, ni saya tak tulis. Okay, so how are you going to know? Okay, how are you going to know which strand that we going to transcribe? Sebab kadang-kadang soalan tak bagi. Okay, soalan tak bagi. So, yang mana satu kita nak transcribe?
3 prime to 5 prime.
Alhamdulillah. 3 prime to 5 prime, why? Why? Can
Sebab apa tu? Apa nama dia, boleh baca daripada 5 prime to 3 prime.
Ya, betul tu. Siapa? Adele kan? Okay. Sebab kita akan copy strand yang ini. Macam Falih cakap, dia akan copy DNA template strand. Okay, DNA template strand. Dia akan copy strand yang ni. Katakanlah soalan tak bagi DNA template strand, awak mesti tahu dia akan copy, dia akan transcribe strand yang ini. 3 prime to 5 prime. Yang ni yang dia akan transcribe. Yang atas ni dia tak transcribe. Kenapa dia tak transcribe? Sebab RNA polymerase only can work from 5 prime to 3 prime. Okay? DNA polymerase only can work from 5 prime to 3 prime. That's why you can see that mRNA dia punya strand adalah 5 prime, 3 prime. Tentu. Okay? Alright? So I repeat, we only going to transcribe, in transcription, we going to transcribe only one strand, and that is DNA template strand, which is 3 prime to 5 prime. Why? Because there are two reasons. First reason, RNA polymerase only synthesize from 5 prime to 3 prime. Second reason, we going to produce mRNA that is 5 prime to 3 prime. Okay, so yang atas ni kita tak guna. Boleh ke? Okay, boleh angkat tangan? Mungkin suara segak-segak. Okay, yang lain? Boleh ke? Alright. Okay, so that is transcription. Okay, so if you see this strand, if you see the DNA template strand, when we going to transcribe, that's why dekat sini bila kita transcribe, bila kita baca, T complementary to A, G, C, A, U, C, G. Bila kita nak baca the DNA template strand, make sure you must know the basic things, the basic concept. For RNA, A is complementary to U, G complementary to C. That is the basic concept of nucleic acid. Okay, so katakanlah kita baca yang ni. C, 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 G, A, G. Kita tahulah C complementary to G. G, 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 C, U, C. Okay, baca. Okay, soalan akan bagi DNA. Okay, mungkin ada soalan dia akan bagi DNA and they ask you to transcribe what is mRNA strand. Okay, and along the mRNA, we have codon. Okay, dekat mRNA ni kita ada bases ni kan. Okay, bases ni kita panggil sebagai codon. And codon is read in triplet base. Codon ni kita baca dalam secara triplet, secara kembar tiga. Secara kembar tiga, satu kembar tiga kita panggil sebagai satu codon. And codon, what you need to know is we have start codon and stop codon. Start codon is AUG, always AUG. Okay, start codon always AUG. Stop codon we have three. We have UA, UA, UGA. UA, UA, UGA. Okay, UA, UA, UGA. Okay, stop codon kita ada tiga, UA, UA, UGA yang ni. Stop, stop, stop. Kalau dia jumpa salah satu stop codon, the process of translation will be stopped. Okay, once it reach stop codon, translation process will be stopped. For example, if you refer to this diagram, if you refer to this slide, so we have AUG. AUG is the start codon. So bila dia adalah start codon, we going to read from this region. Okay, we going to read from this start codon. So for example, start codon. So kita nak tahu start codon ni apa amino acid dia? Apa amino acid dia? How you going to determine the amino acid? Okay, you going to refer to this table, genetic code table. Okay, genetic code table. To find amino acid, you need to refer to genetic code table. So for example, I have AUG. So I refer to the genetic code table. So AUG codes for amino acid methionine. Okay, so I write methionine dekat sini. Okay, I write methionine. Next, we have AGC. Next codon, we have AGC. So kita carilah mana AGC. Cari kejap AGC, apa dia punya amino acid? AGC. First letter A. Second letter G. So mana? Serine. Yes. Serine. Serine is our amino acids. So our amino acids for AGC is serine. So awak akan baca setiap satu ni, awak baca sampailah you reach, okay, until you reach stop codon. Once you reach stop codon, there is no amino acid. You can see that, okay, we have UAA here. UAA is one of the stop codon. If you refer to the genetic code table, there is no amino acid for stop codon. Sebab itulah kita tak ada, kita kosong kan kat sini. Dia tak ada dah stop codon. So your polypeptide chain will be this one. Ini adalah awak punya polypeptide chain or protein. Okay, boleh ke?
Madam,
Ya,
genetic code table ni kita kena hafal ke?
tak perlu. Genetic code table, good question Iman. Genetic code table tak perlu hafal. Soalan akan bagi genetic code table. Okay, dalam biomix awak pun, dalam pasir awak pun dibagi genetic code table. So you don't have to hafal. Okay, alright. So that is codon. Okay, codon. We also have anticodon or anti-codon. Okay, depends on how you pronounce it. Make sure you spell correctly. So the codon, the anti-codon, the anticodon is located at tRNA. Sama juga kita baca sebagai 3, tapi anticodon ni dia duduk dekat tRNA. So kita tengok tRNA. This is tRNA. Okay, this is tRNA. This one. These two diagram. Okay, these two diagram is tRNA. You can see that this is where anticodon is located. Okay, anticodon is located. Dia duduk dekat bawah. Anticodon will be complementary to codon. Okay, anticodon will be complementary to codon. And at this side, okay, kat kepala dia ni, at kepala of tRNA, there is amino acid attachment site. Okay, attachment site. Kalau kita tengok gambar, kalau gambar awak jumpa, ni adalah tRNA. Okay, that is tRNA. tRNA kalau kita lukis, contoh saya lukis macam ni lah. Kepala ada amino acid, kaki ada antikodon. Okay, itu adalah tRNA. Dan dalam gambar pun dia lukis macam tu. Okay, for example, ada tak lagi? This is tRNA. Kepala ada amino acid, kaki ada antikodon. Antikodon is complementary to codon. For example, if the codon is AUG, the anticodon will be UAC. Okay, so for example, if I have codon, okay, katakanlah saya ada codon GCT. Okay, this is my codon. Okay, this is my codon. What is my anticodon? Anticodon saya apa? Kalau codon saya GCT. Betul lah. Kalau codon saya adalah GC, bukan T, GCA. Sorry. Okay. If my codon is GCA, what is my anticodon? Apa dia?
CGT.
CG?
T.
CGT. Bukan CGT sebab kita adalah RNA. Kalau RNA, A kena complementary dengan U. Ya? Betul tak? Kan? Okay. So bila kita GC, sebab ni ingat kalau translation kita discuss pasal RNA. Okay, kalau every translation punya proses kita cakap pasal RNA saja. So bila RNA kita guna basis A, U, G, C. Tak ada huruf T. Okay, tak apa. It's okay. It's okay to make mistake. Saya pun buat mistake tadi. Kita belajar. Alright? So that is concept of codon dengan anticodon. Boleh ke? Boleh ke?
Boleh.
Okay. Alright. Then okay, I already explained about this one, right? The RNA splicing. Dah ke?
Dah.
Dah semalam. Okay, bagus. Thank you. Okay, next after genetic code table, after genetic code and everything, we're going to proceed for the next process. Okay, kita dah habis dah proses transcription. Okay, we move on to the next process, which is translation. Translation is translating the genetic code. We're going to translate the genetic code carried by mRNA to synthesize protein. Okay, kita translate. Awak pernah guna Google Translate ke?
Nah, ada.
Pernah. Okay, sama juga. Katakanlah awak nak translate macam saya, katakanlah konsep Google Translate tu kita nak translate bahasa yang kita tak faham. Contoh saya nak translate bahasa apa? Bahasa French, bahasa Perancis contoh. Saya nak translate ke dalam bahasa Melayu atau bahasa Inggeris. So I'm going to translate something that I don't know into something that give makna, okay, bagi membawa satu maksud. Sama juga protein synthesis translation ni. Translation is the process of translating the genetic code. Kita nak translate genetic code, which is codon tadi, yang A, G, C, 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 C itu, huruf-huruf tu kita nak translate kepada protein. Kita nak translatekan dia kepada protein. And in order to do that, we need three things. We need mRNA, tRNA, and ribosome. Okay, we need mRNA. Along the mRNA, we have codon. Okay, along the mRNA, we have codon. Then we have tRNA. tRNA, we have anticodon and also amino acid. Okay, and the last one, we have ribosome. Okay, I already explained you about tRNA. Okay, function. Okay, tRNA, yang ni saya cakap pasal tRNA tadi lah. Function of tRNA, it will transfer amino acid from cytoplasm to a growing polypeptide chain in a ribosome. Okay, attachment of amino acid to its tRNA is called amino acid activation, which is catalyzed by amino acid tRNA synthesis. So in translation process, okay, in translation process, there are two enzymes that you need to know. The first enzyme is this one, amino acid tRNA synthesis. This amino acid will be used during amino acid activation. Okay, the first enzyme is amino acid tRNA synthesis, and this enzyme will be used in amino acid activation. Before tRNA, okay, this is tRNA. Before tRNA proceed, okay, before this is tRNA, before tRNA can proceed to the process of translation, this tRNA must undergo amino acid activation, which is catalyzed by amino acid tRNA synthesis. So what happened here during amino acid activation? Okay, this tRNA yang ni tak ada kepala. Okay, tak ada kepala, tak ada amino acid dekat kepala dia. But after process amino acid activation, tRNA akan ada kepala. And that kepala is amino acid. Okay, so if you refer to the next slide, this is process of amino acid activation. So you can see that this is amino acid tRNA synthesis. This is amino acid. Okay, amino acid tRNA synthesis is the enzyme, right? This enzyme. So what happened here? This amino acid, it will bind to the enzyme. Okay, it will bind to the enzyme. And this is tRNA. Nampak tak? tRNA tak ada kepala kan? Kan?
Tak ada.
Tak ada. Okey, kepala tu apa dia? Apa maksud kepala tu? Kepala tu apa dia?
Amino acid.
Yes, kepala tu adalah amino acid. Okay, so if you refer to this diagram, if you refer to this slide, amino acid dah masuk dulu. Then tRNA will bind to the amino acid tRNA synthesis, and this amino acid, this amino acid tRNA synthesis will catalyze the attachment of amino acid to tRNA. Maksudnya amino acid tRNA synthesis ni dia akan gabungkan. It will catalyze the attachment. Maksudnya dia akan tolong attach amino acid to the tRNA. So bila dia masuk tak ada kepala, bila dia keluar ada kepala. Okay, masuk tak ada kepala, bila dia keluar ada kepala. Bila dah ada kepala tu, kita tak panggil lagi sebagai tRNA. No, we don't call that as tRNA. We call that as amino acid tRNA. Okay? Boleh ke setakat ni angkat tangan kalau boleh?
Madam, cuma nanya tu.
Ya.
Yang amino acid ni dia daripada mRNA.
Amino acid memang ada dekat cytoplasm.
Yang mRNA tadi kan yang amino acid dia ambil daripada nanti madam waktu akhir lah.
Okay. Alright. Yang lain okay? Okay, diam. Okay, so itu kita dah cerita pasal amino acid activation. Next kita tengok pada ribosom. Ribosom ni dia ada dua tempat, dia ada dua components. Yang komponen pertama adalah small subunit. Okay, small subunit. At the small subunit we have mRNA binding site, and then we have large subunit. At the large subunit we have three sites. We have E site, P site, and also A site. Okay, kita ada E site. E site for exit. Okay, E site is for exit, E, exit. P is P site. Okay, P site is peptidyl tRNA binding site. P site is the one that hold tRNA carrying the growing polypeptide chain. P site ni lah yang dia akan pegang tRNA yang kepala dia ada polypeptide chain. A site is amino acid tRNA binding site. This is the one that hold the tRNA carrying the next amino acid to be added to the chain. Okay, maksudnya kalau kita tengok yang ni, okay, tengok gambar ni. Okay, kalau if you refer to this diagram, this E site is whereby it will discharge tRNA. E site di mana tRNA akan keluar melalui E site, akan keluar melalui exit site. Okay, P site is the tRNA that hold the polypeptide chain. Itu P site. A site is tRNA that carry the next amino acid. Okay, amino acid yang seterusnya yang nak ditambah kepada sini, dia duduk dekat tRNA yang duduk dekat A site. Okay, yang tu. So nanti ini kita ada satu amino acid kan? If you refer to this one, kita ada 4 amino acid. Yang ni ada satu amino acid. Satu amino acid ni lah yang akan ditambah kepada sebelah dia, kepada jiran dia. Okay, itu adalah EP site, E site, P site, A site. Okay. Alright. Then we move on to the stages involved in translation. Okay, we have 3 stages: initiation, elongation, and termination. Initiation adalah start, proses yang mula-mula lah kita start. During elongation, kita ada 3 small step, which is codon recognition, peptide bond formation, translocation. And the last one is termination. Saya akan pergi pada satu-satu. Saya akan explain slow lah. Okay, Allah. Okay, this is the diagram of initiation. Okay, this is initiation. So what happened is, okay, saya cerita dulu, based on ayat. So during initiation, it will bring together mRNA, tRNA, and the 2 ribosomal subunit. Maksudnya dia akan assemble. Okay, dia akan kumpulkan mRNA, tRNA, and the 2 ribosomal subunit. Okay, initiation. That is initiation. So what happens is, small ribosomal subunit binds with RNA. Then, initiator tRNA with the anticodon UAC pair with the start codon AUG. Then, a large ribosomal subunit bind to the small ribosomal subunit. Bila large subunit datang, translation initiation complex is complete. Okay, so by referring to the previous slide, you can see that this is small ribosomal subunit. The small ribosomal subunit, it will bind to the mRNA. Okay, it will bind to the mRNA. Then initiator tRNA. During initiation, kita panggil dia sebagai initiator tRNA. Sebab dia adalah yang start. Okay, dia yang start, initiator, permulaan. tRNA yang mulakan. Okay, this initiator tRNA, it will bind to the start codon, AUG. Okay, which complementary to the base pair, UAC. After initiator tRNA datang, then the large ribosomal subunit bind and translation initiation complex will be complete. Okay, so kalau kita tengok yang ni, GIF ni, you can see that there is initiator tRNA. That initiator tRNA will bind to the start codon, AUG. Okay, bind. Okay, macam tu. Lepas dia bind, barulah large ribosomal subunit datang. Selagi initiator tRNA tak datang, large ribosomal subunit tak akan datang. Okay, so that is initiation. Okay. Next, we have elongation. Okay, elongation. During elongation, we have three small steps. Okay, three small steps, which is codon recognition, peptide bond formation, translocation. Okay, kita tengok dulu codon recognition. Codon recognition maksudnya dia akan recognize the next codon. Okay, codon recognition dia akan recognize the next codon. So what happened during codon recognition? The anticodon of an incoming amino acid tRNA base pair with codon of mRNA in the A site. Okay, kita tengok yang ni. Ini adalah UAC kan? The next codon adalah UAC. Betul tak?
Betul.
Betul kan? So next codon adalah UAC. So siapa yang datang nextnya? Ini adalah proses codon recognition lah. Dia akan recognize the next codon. So UAC. So next tRNA, okay, yang bawa amino acid apa? UAC, apa dia punya amino acid? Tolong tengok kejap genetic code table. UAC, apa dia punya amino acid?
Tyrosin.
Tyrosin Okay, tyrosin TYR. So amino acid TYR. Okay. If I have codon UAC, what will be my anticodon?
AUG.
AUG, right? So AUG. AUG is my anticodon, sorry. Okay, AUG is my anticodon. Faham je lah. Okay, AUG is my anticodon. So tRNA yang ni, this amino acid tRNA, it will occupy the A site. Okay, that is codon recognition. So katakanlah kita tengok balik, codon recognition kita recognize the next codon. Okay, saya patah balik, tengok dia masuk. Masuk, then keluar. Pergi sebelah. Masuk, peptide bond formation, then pindah sebelah, masuk the next. Okay, itu adalah codon recognition. Then we have peptide bond formation. Peptide bond formation, during peptide bond formation, there is second enzyme that you need to know, which is peptidyl transferase. Okay, peptidyl transferase. Peptidyl transferase is the enzyme that catalyze the formation of peptide bond. Okay, so kat mana dia akan catalyze? Dia akan catalyze the formation of peptide bond between amino acid in the A site and the growing polypeptide in the P site. So for example, okay, peptide bond, kan, tu dipindah sebelah. Next tRNA datang, peptide bond, pindah. Okay, so saya repeat balik. Peptide bond and then yang ni akan pindah kepada tu. Okay, A site, amino acid. Dia akan form peptide bond between amino acid in the A site and the P site. Okay, next, between valine and arginine. A site, P site. That is peptide bond formation. Okay, this one. Kita tengok lagi. Peptide bond formation, peptide bond. Between amino acid in the A site and the P site. Bila ada peptide bond, barulah kita boleh hasilkan polypeptide chain or protein. Okay, boleh ke? Boleh ke setakat ni? Guys, boleh ke?
Boleh.
Alright, yang lain okey ke?
Boleh.
Okey, Alhamdulillah.
Okay.
Next, we move on to translocation. Okay, translocation maksud dia akan translocate. Okay, pindah lokasi. So what happened here, the ribosome will translocate tRNA in the A site into the P site. So you can see that from A site, it will go to the P site. Okay, nampak? A site, it will go to the P site. Okay, Tengok lagi. After peptide bond formation, there is process translocation. From the A side, it will move to the P side. Translocate. P side. Actually, dia tak tunjuklah dalam gambar ni. Tapi lepas yang ni, dia bukan terus keluar macam tu saja. Dia akan masuk ke dalam E side. Sepatutnya GIF ni ada satu lagi lah, E side. Sepatutnya dia pindah ke dalam E side dulu. Contoh kan, yang ni. Dia akan masuk ke sini dulu, E side. Cantum, dah tak ada kepala tu, barulah dia akan pindah kepada E side. Okay, kalau kepala dia dah tak ada, okay, meteor ni tadi ada kepala, tak ada kepala. Dan dia akan pindah masuk ke dalam E side. That is translocation. A pindah kepada P pindah kepada E. Okay, translocate. Siapa yang pindah? tRNA yang pindah. Right? Next is termination. Okay, the last stage. We already discussed initiation, elongation. Next is termination. Termination occur when it reach one of the stop codon. Stop codon are uwa, uga. Kan? Stop codon kita adalah uwa, uga. Jumpa je salah satu. Okay, when ribosome reach stop codon, release factor will enter A site. Okay, once ribosome reach stop codon, release factor will enter the A site. Okay, and the release factor ni function dia buat apa? This release factor, it will hydrolyze the bond between tRNA in the P site and the last amino acid of the polypeptide chain. Meaning that this release factor, it will hydrolyze bond yang ini. Ni, bond yang kali merah, nampak kali merah. Okay, it will hydrolyze the bond between amino acid, between the last amino acid and tRNA in the P site. Dia akan putuskan. Okay, release factor ni macam scissor. Dia akan hydrolyze, dia akan potongkan last amino acid dengan tRNA. Okay, so what happen is the polypeptide sekarang dah boleh free. Bila kita dah putuskan, polypeptide chain ni dah free. Polypeptide chain tu nak pergi mana? Protein tu nak pergi mana lepas dia free? Soalan kebat, soalan pernah tanya. Soalan kebat ni dia pernah tanya. What happened to the free polypeptide chain? What happened to the protein after it is released by release factor? Siapa? Dia nak siapa? Dia nak pergi mana?
Golgi body.
Kenapa Golgi body? Kenapa Golgi body?
Dia nak package dengan transport.
Apa dia? Siapa kita tahu jawapan tadi? Adil. Kenapa nak pergi Golgi body, Adil?
Sebab dia nak packing apa semua tu dulu baru hantar keluar sel.
Okey, Falih, apa jawapan awak? Falih.
Lebih kurang juga lah. Dia nak package dulu. Dia nak modify dulu kalau dia perlu modify. Kalau tak pun tak apa, dia lepas tu baru dia hantar ke mana yang dia nak ambil.
Okey, Atira, jawapan awak apa? Ke mana dia akan pergi? Next.
Golgi body juga sebab nanti yang tu dia akan nak bawa gelak pada 19.
Pandai, tiga-tiga betul. Okay, so what happen is this polypeptide chain, this protein, it will go to Golgi body or Golgi apparatus, because at the Golgi body, this is when the protein will be modified, packaging, sorting, and shipping. MPSS. Okay, that polypeptide chain, that protein will be modified, packaging, sorting, and shipping. So the next location at the cytoplasm, this polypeptide chain will go to the Golgi body. Okay. Next, after polypeptide chain is free, then the two ribosomal subunit and the other components will dissociate. Okay. Lepas polypeptide chain dah free, pergi mana-mana dia suka, tak ada lagi mana dia suka, dah pergi free, nak pergi kepada Golgi body, then the translation kompleks yang ni lah, it will dissociate. Habis dah kerja dia. Okay, dia akan dissociate. That is termination. Alright? Okay. So ini sama je lah. So kalau you tengok, tengok gambar ni, release factor masuk. Sekejap Okay, dia dah jumpa stop codon, which is ugar. Release factor will enter the A site and then it will hydrolyze the bond and translation complex will be dissociate. Okay, I repeat again. Okay, once it ribosome reach stop codon, release factor enter the A site and then it hydrolyze the bond, then it will dissociate. Okay, next is polyribosome. Saya discuss sampai sini saja. Okay, polyribosome nak cerita pasal ni and then saya tunjuk video dan habis. Polyribosome is ribosome lah. Poly ni banyak. Okay, banyak. Polyribosome is a structure of many ribosome attached to single mRNA. Okay, dia refer kepada banyak ribosome attached kepada satu mRNA. You can see in this diagram, we have 4 ribosome and it attached to only one mRNA. Satu mRNA saja dia attached. Why we have polyribosome? To accelerate the synthesis of many polypeptides of the same kind. Soalan pernah tanya 2 markah. Dululah masa soalan keluar masa topik ni keluar final exam, dia pernah tanya 2 markah. Kalau dia tanya dalam MCQ, dalam UPS, what is polyribosome? Or dia boleh tanya, what is the significance of polyribosome? Okay. So, the significance of polyribosome to accelerate the synthesis of many polypeptides of the same kind. For example, we want to produce polypeptide yang banyak. Satu polypeptide. Kita nak polypeptide B, contoh. This polypeptide. Kita nak polypeptide yang nama dia B ni. So, B ni kita tak nak satu je. Kita nak hasilkan banyak polypeptide. So, macam mana kita nak buat? Kita ada polyribosome. Okay, kita nak mempercepatkan sintesis of polypeptide nama dia B ni, kita ada this mechanism, okay, this process, polyribosome. Boleh ke? Ada soalan setakat ni? Soalan ke? Faham ke? Tak? Faham ke?
Boleh.
Boleh Okay, saya tunjuk satu video mengenai translation and then kita habiskanlah kelas kita sikit je lagi. Alright, so I already assign you quiz kan? Quiz topik 5, population genetic. Ada yang dah hantar, baru 4 orang kot hantar. Okay, baru 4 orang hantar ke baru 3. So tolonglah buat, lepas tu saya dah assign add puzzle and saya juga dah assign quizzes. Nanti saya akan tambah lagi quizzes dengan add puzzle. Sebab soalan ni, sebab ini adalah memang dia adalah assignment. Tapi dia perlukan banyak latihan lah. Okay, kita tengok yang mana. Inilah. So this is process of protein synthesis. Dah keluar ke belum? Sebentar. Okay. Dah. Okay. Okay, so that is process transcription lah, DNA. Okay, kita start dengan proses transcription. That is DNA, dekat DNA ada gene. Okay, along our DNA kita ada gene. Gene itulah yang kita nak transcribe. So, kita ada promoter and terminator. Okay, promoter and terminator. So, that is the region that we going to transcribe. So, what happened first? We have protein. Protein ni lah yang kita nak transcribe dulu. So, kita nak transcribe dulu, kita akan start dengan proses transcription, which occur in the nucleus and translation in the cytoplasm of the cell. So what happen is, this is the yang besar ni adalah RNA polymerase. This RNA polymerase, okay, it will bind and that is translation lah. Dia cerita dulu. Okay, transcription. Transcription, that is the DNA, and we're going to transcribe only that region. So we have RNA polymerase. RNA polymerase, it will bind to the promoter. Okay, it will bind to promoter during initiation. So during initiation, you can see that it will bind to the promoter region. So when it bind, okay, it will unwind the double helix. Okay, it will unwind the double helix. Maksudnya dia akan buka dia punya double helix. Okay, dia akan buka menjadi bentuk macam ni, replication bubble. Okay, so during elongation, okay, this is dah start dah proses elongation. This RNA polymerase will catalyze, okay, catalyze the addition of RNA nucleotide complementary to the DNA. Dia tambah je, RNA polymerase tolong tambah until it reach terminator. Once it reach terminator, the transcription process will be stopped. Okay, will be stopped. That mRNA, the mRNA will be released. Okay, mRNA will be released, and in eukaryotic cell, mRNA will undergo process called RNA splicing. We have exon region, intron region. Kita tak nak intron. Kita tak nak mana Tak nak mana? Kita tak nak yang mana? Kita tak nak sekejap. Kita tak nak intron. Okay, kita tak nak intron. Kita nak exon. Okay, kita tak nak intron, kita nak exon. So this is spliceosome. Spliceosome will buang. Okay, it will remove the intron. It will remove the intron, and we only have mRNA that has exon. The exon, the coding region. Then the mRNA will exit the nucleus through nuclear pore, and then it will go to the cytoplasm for the process of translation. So along the mRNA, we have codon. Okay, codon, triplet bases known as codon. So to start the process of translation, codon lah triplet bases GCA. So kita nak macam mana kita nak baca amino acid, okay, kita kena refer kepada genetic code table. Okay, so that is start codon AUG, stop codon UA. Okay, so this is that is small ribosomal subunit. Small ribosomal subunit bind to the mRNA, and then the initiator tRNA, which bring amino acid methionine, will bind to the start codon. Okay, it will bind to the start codon. After it bind to the start codon, then the large ribosomal subunit. Okay, barulah large ribosomal subunit datang. And that initiator tRNA will occupy P site, bilik yang tengah. Okay, translation complex will be completed. Okay, that is initiation. Okay, E site, P site, and A site. Alright, during elongation, it starts with codon recognition. We're going to recognize this codon. Okay, kita akan recognize codon yang ni. Okay, kita tengok codon yang ni. So next tRNA, the next amino acid tRNA, okay, it will bind to the next codon. Okay, it will bind to the next codon. After it bind to the next codon, okay, after it bind, that is amino acid. After it bind to the next codon, this amino acid will be transferred to the bukan sini, ini ke sini. Okay, this amino acid will be transferred to the P site. Okay, to the next amino acid catalyzed by peptidyl-transferase. So boleh nampaklah tadi, okay, yang ni, yang tak ada kepala tu, ini tRNA tak ada kepala tu, dia akan pergi gerak kepada E site. Dekat E site itulah baru dia keluar. Okay, baru dia keluar. Okay, next amino acid, okay, tambah lagi keluar. Sampailah, okay, tambah polypeptide, sampailah banyak-banyak macam ni. Until it reach stop codon. Once it reach stop codon, release factor will enter the A site, and then it will hydrolyze the bond between last amino acid and the tRNA. Then translation complex will be dissociate. Dapatlah kita polypeptide chain or this protein. So kita dapatlah polypeptide chain and protein yang kita nak daripada DNA tadi. Bayangkan daripada DNA kita dapat protein yang kita nak. After that, it will go to the Golgi body. This is Golgi body to be modified, to be packaging, to be sold, to be shipping. Nak guna kepada luar sel ataupun nak guna ke dalam sel. So katakanlah nak guna kepada luar sel, dia akan keluarlah. Okay, keluar daripada sel tu by the process of exocytosis. Okay? Boleh ke guys? Ada soalan? Ada soalan tak?
Tak ada ni.
Tak ada. Clear ke? Faham ke yang lain? Siapa tak faham? Siapa tak faham angkat tangan. Okay, so saya assume awak faham. Nanti tolong buat app puzzle, quizzes dengan quiz tu lah. Okay. Kalau rajin kan kalau rajin. Kalau ada masa lebih ke awak buatlah. Buat apa benda? Buat, sekejap buat apa ni? Buat biomix, soalan pasir biomix. Okay, kalau awak tengok topik 6 biomix memang ada soalan MCQ saja. Soalan structure essay tak ada sebab dah tak ada dalam exam. So ada MCQ sahaja. And jangan lupa hantar draft next week. Hantar draft assignment next week. Okay, hari Jumaat. Okay, awak boleh hantar start pada Isnin lah ataupun boleh hantar dalam minggu ni. Okay, alright. Kalau tak ada apa-apa soalan, saya stop sampai sini saja. See you on next Wednesday lah. Jap. Sebentar. Apa dia? Kak Ipin nak cakap apa?
What the draft assignment?
Ya. Huh?
Not how to do the draft assignment.
How to do the draft assignment? What do you mean? You have to do.
Upload the whole Wakelet to you.
Yeah. Okay. You do the workload first. Okay, and then give me your ID. Give me your username. Ataupun you hantar secara PDF file to me via WhatsApp. Then I will check. Boleh ke?
Means we don't need to insert all the information into the Wakelet first.
Masukkanlah semua information dulu baru saya boleh check draft. Kalau tak saya nak check apa? Kan? You kena hantar dulu, you kena masukkanlah dulu information yang you cari. Lepas tu baru hantar. Barulah suruh saya check draft. Macam mana saya nak check draft tanpa you punya workload tak ada apa-apa? Contoh ada cover image saja. Macam mana saya nak check? Cover image je, buat apa saya nak komen cover image? Contohlah. Okay? Check draft maksudnya awak dah ada info mengenai, awak kan dah ada info, awak dah buat at least part B, part C, part D tu dah buat. Okay? Yang lain tu tak apa. Yang lain tu tak apa. Tapi kalau boleh buat semualah, first draft tu mesti ada semua. Mesti dah ada information segala benda dalam tu. Okay? Mungkin awak tak nampak. Macam mana cakap? Sebab akan ada mistake. Okay, akan ada mistake. Contoh gambar lah. Gambar selalunya student buat mistake. Bukan soalan. Saya nak eukaryotik, awak bagi soalan untuk, awak bagi gambar untuk prokaryotik. So, saya akan check benda-benda macam tu lah. Right? And untuk minggu depan, hari Rabu, ya Kak Peng?
You cakap dulu.
Okay, untuk minggu depan hari Rabu tu awak punya eksperimen kan 6. Eksperimen 6. Basic technique in isolating DNA. So, untuk Rabu minggu depan, kita tak buat kelas awak buat eksperimen tu. Okay? 2 jam minggu depan pada hari Rabu, kita tak buat kelas tapi awak buat eksperimen tu. Yang penting hari Khamis minggu depan, semua dah hantar video. Dah hantar video melalui YouTube. Okay, video YouTube yang awak dah buat eksperimen 6 isolating DNA tu. Okay, so untuk isolating DNA awak boleh start buat minggu ni ataupun awak boleh buat minggu depan pada hari Rabu yang kita tak ada kelas tu. Okay, hari Rabu tu kita ada kelas tu, awak buatlah eksperimen tu selama 2 jam. Okay, clear?
Clear.
Clear.
Okay, cuba balik pada hari Khamis. Ya, Kak Peng nak tanya apa?
Oh, the information in notes, can we straight copy or have to change?
Boleh copy, tak ada masalah.
Oh, okay.
Okay. Alright, kalau yang lain tak ada apa-apa soalan, kita stop sini saja. Tapi kalau masa you tengah buat draft tu, kalau apa-apa you tengah buat soalan quiz ke, ada apa-apa soalan, boleh message saja. Right? Okay guys, bye-bye, take care, have a nice weekend, see you next week. Assalamualaikum, bye-bye.
Kesalam.
Thank you, madam.
Thank you guys, thank you.
Thank you.